Gene therapy. Gene technology and our rapidly increasing knowledge of the positions of particular genes on our chromosomes had given us the opportunity to identify many genes that are responsible for genetic disorders such as sickle cell anemia and cystic fibrosis. Okay, now these are the diseases that are caused by genetic disorders in our cells. Now, if we do not know the genes that are responsible and the location of the genes in the genome, we cannot know which gene is responsible for the disease. And if we do not know the problem, then we cannot find a solution for that. So, for both finding the problem and also finding a solution, we need to know the location of the genes and which genes are present. And since we know this using genetic technology, it really helped us to accelerate this process. Now, when genetic engineering really began to get going in the 1990s, it was advised that it would not be long before gene technology could cure these disorders by inserting normal alleles of these genes into the cells. Since these diseases are caused by the faulty genes, if we can insert the normal alleles or normal genes, alleles are alternative forms of genes, so same thing, into the cell, then that problem could be cured, or at least the effects of the disease can be minimized. This process is called gene therapy. The gene therapy has proved to be far more difficult than was originally thought. The problem lies in getting normal alleles into the genes of the normal alleles of the genes into a person's cells and then making them work properly when they get there. Okay, that's said. The most common vectors that are used to carry the normal alleles into host cells are viruses, often retroviruses or lentiviruses, or small pairs of phospholipid called liposomes. Occasionally naked DNA is used. They usually do not use plasmids. We have discussed how plasmids can be used to insert the genes into a bacteria, and in this case it will be inserted into a cell. The first successful gene therapy was performed in 1990 on a 4-year-old girl from Cleveland, Ohio. She suffered from the rare genetic disorder known as severe combined immunodeficiency. In this disorder, the immune system is crippled and sufferers die in infancy from common infections, because we know that immune systems does the process of defeating the invading pathogens. But if the immune systems are destroyed or does not work because of the faulty gene, then even normal infections can have an effect on our body, and this usually sadly the people suffering from this condition die when they're infants. Children showing the condition are often isolated inside plastic bubbles to protect them from infections, so that no pathogens can enter the baby's body through the plastic bubble. The defect in SCID involves the inability to make an enzyme, adenosine deaminase, which is vital for the functioning of the immune system. Okay, some of the child's T lymphocytes were removed and normal virus alleles of the ADA gene were introduced into them using a virus as a vector. So T lymphocytes produce this ADA enzyme, and since the genes of the T lymphocytes are changed, that's why they cannot produce this enzyme. But if few or more T lymphocytes were collected from the body of the baby and the gene inserted, so those lymphocytes will produce the enzyme, and thus the immune system can work quite a lot effectively, which would be zero if they were not produced. Okay. Okay. The cells were then replaced. This was not a permanent cure. Regular transfusion every 3 to 4 or 5 months were necessary to keep the immune system functioning, because these are T lymphocytes and they cannot transform that gene into their offsprings, because well, it's an advanced topic, but most probably the extra genes that are added are not actually added into the like complete genome that will be like divided in mitosis, and that is why they cannot be passed on to the daughter cells, and that is why they need continuous injections of administering of these T lymphocytes, which will produce the enzyme, because T lymphocytes die. So yes, as even though the T lymphocytes can replicate because they have a large nucleus, they cannot transfer that normal allele into their daughter cells. Okay, 2 years later, gene therapy using stem cells harvested from bone marrow was successful. But in France, in 2000, 4 children who had received gene therapy for X-linked SCID developed leukemia as a result of using retroviruses as a vector. So, if the stem cells were like in the bone marrow were harvested, and they can be inserted the gene, then we know that stem cells are formed into the red blood cells and white blood cells. So then the white blood cells produced from those stem cells may have will have the normal allele of the gene, but there is a problem, and that is the 4 children who had received gene therapy for X-linked SCID developed leukemia as a result of using retroviruses as vectors. Leukemia is when leukocytes are not that much present. Why does this happen? Retroviruses insert their genes into the host genome, but they do so randomly. This means that they may insert the gene within another gene, or more dangerously, into the regulatory sequence of a gene, which may then activate a nearby gene causing cancer. Yes, since they do it randomly, they may not insert it into the position where we want it to. It may insert it into an unwanted position, because retroviruses does the insertion of the genes randomly, and if by any chance they put the gene where they'll activate a nearby gene causing cancer, then that will be very sad. Since researchers have used lentiviruses as vectors, they also insert their genes randomly into host genomes, but they can be modified to inactivate replications. See, but if we use lentiviruses, then the lentiviruses can be modified to suppose like these lentiviruses have a stop switch, but retrovirus will keep doing it. So if we have a stop switch, we can regulate the insertion of the genes. Okay, these also insert their genes randomly into host genome, but they can be modified to inactivate replication. HIV has been disabled in this way to act as a vector. The adeno-associated virus is also now used as a vector. This virus doesn't insert its genes into the host genome, so they are not passed on to daughter cells when a cell divides. So this is a problem when host cells are short-lived, but the virus has been used successfully with long-lived cells such as liver cells and neurons. So the adeno-associated virus is also used as a vector, but the problem is the virus doesn't insert its gene into the host genome. Like maybe they are just keep it beside the genome, but transcription and translation occurs somehow, So since they cannot be inserted into the host genome, they won't be like transferred into the child or daughter cells. Since, say for example, let me explain you by drawing, it will be much easier. Oops. Okay, say for example, this is the nucleus, like the genes, but if we insert then this is the gene that we want to insert. If we insert it, it won't be inserted into the genome, it will be outside. Thus, when the mitosis occurs, the daughter cell will have this genome, and the father cell will have its older genome and the gene which was inserted, and so they can be inserted into the daughter cells. It only stays in the father cells, and since they are genes, they are not they do not live forever. They cannot produce the product for forever, so they need continuous insertion of the genes into many cells so that they can produce the product. So that's a problem. But for long-lived cells, yeah, it could have occurred because those cells lived for a long time. This work on vectors had led to increasingly successful gene therapies in the last few years, including the following: the eyesight of young men with a form of hereditary blindness, Leber congenital amaurosis, in which retinal cells die off gradually from early age, has been improved. Okay, so these examples are basically how gene therapy helped people. We will look at more detail in genetic disorders, cystic fibrosis, to illustrate some of the problems facing gene therapy. So basically, what is gene therapy? If I tell you, you will say that a genetic disorder occurred because of a change in the genome, and thus we will take few of those cells, insert the normal alleles of the gene or the genes, and then we will insert the cells so that they can function normally, but they won't live forever. Once they die, we need to do it again. Okay, so cystic fibrosis. Cystic fibrosis is a genetic disorder in which abnormally thick mucus is produced in the lungs and other parts of the body. Okay, a person with cystic fibrosis is very prone to bacterial infection in the lungs because it is difficult for the mucus to be removed, allowing bacteria to breed in it. So mucus is actually a mixture of mucus. Yes, of course, mucus is a jelly-like substance which is produced in the lungs to take in the bacteria and debris, dust from the air inhaled. Okay, so they need to be removed, else it's a good environment for bacteria to breed in. Usually we have cilia to remove it, but the mucus of the cystic fibrosis suffering person is abnormally thick, so not even the ciliary movement of the cilia or even coughing can remove it, which is very dangerous. People with cystic fibrosis need daily therapy to help them to cough up this mucus. So they need therapy kind of thing, they vibrate the back, and so the mucus can be coughed up. Okay, the thick mucus adversely affects many other parts of the body. The pancreatic duct may be blocked, and people with cystic fibrosis often take pancreatic enzymes by mouth to help them digestion, because pancreatic juice cannot be secreted into our body. That is because it is blocked. That's why we need to take the pancreatic enzymes by mouth through pills or medicines. Around 90% of the men with cystic fibrosis are sterile because thick secretion blocks ducts in the reproductive system. If the ducts in the reproductive system are blocked, then sperm cannot be ejected because they are blocked. And so how can they reproduce if there are no sperm, because in reproduction, sperm and eggs, the man's sperm and the woman's egg join to form a zygote. That can't happen. Cystic fibrosis is caused by a recessive allele of the gene that codes for the transporter protein called CFTR. We know what's a transporter protein. It is a protein in the surface of cells, cell surface membranes, which is responsible for the entry and exit of substances. This protein sits in the cell surface membranes of the cells, in the alveoli, and also everywhere in the body, and allows chloride ions to pass out of the cells. Recessive allele codes for a faulty version of this protein that does not act properly as a chloride ion transporter. Normally, the cells lining the airways and in the lungs pump out chloride ions through the channel in the cell surface membrane formed by CFTR. This results in relatively high concentration of chloride ions outside the cells. This reduces the water potential below that of the cytoplasm of the cell. So water moves out of the cell by osmosis down the water potential gradient. It mixes with the mucus there, makes it thin enough for easy removal by sweeping movement of cilia. Okay, let me explain you by drawing. Say for example, this is the alveolar wall, and this is the inside, and this is the outside. Here is the mucus, which needs to be removed. It is very thick. So usually what happens, if the water potential, if we move the chloride ions here, then what happens? Then the water potential here is very low water potential, but inside the cell is high water potential. First of all, the chloride ions need to be pumped out by the CFTR proteins from inside the cells, by CFTR proteins from inside the cells. Thus low water potential, high water potential. Then what will happen? This water will move from the higher concentrated area to the lower concentrated area. Then what will happen? Then which color do I use? This mucus here will be thinner, and thus, since they are thinner, they can be easily removed from the lungs, from the alveoli, by the sweeping movement of the cilia. But since this cannot happen in the body of a person with cystic fibrosis, the thick mucus is not removed, not even by coughing, which is very sad. Yeah. However, in someone with cystic fibrosis, much less water moves out of the cell, so the mucus on the cell surfaces stays thick and sticky. The cilia or even coughing can remove it. See, this is the transporter protein in the cell membrane, and it causes the chloride ions to move out of the cell, and then what happens, I've just explained you. Now let's look at the CFTR gene. The CFTR gene is found on chromosome 7 and consists of about 250,000 bases. Mutation in this gene have produced several different defective alleles. Okay, defective alleles. The recessive one is actually the defective in the practical case. The commonest of this is the result of a deletion of 3 bases. 3 bases makes one amino acid. One amino acid being absent into the protein. Thus the shape of the protein change, the identity of the protein change, the how the protein works change, everything of the protein is changed. Thus the protein cannot do its normal work, and that is why it happens. The CFTR protein made using the code on this allele is therefore missing one amino acid. The machinery in the cells recognizes this, that this is not the right protein and does not place it in the cell surface membrane. So one of the mutations that causes the cystic fibrosis causes the deletion of the amino acid, and thus the cells do not at all identify this as the protein they need, and so they do not place the protein. The cell surface membrane do not have the protein. While in another example we have learned that it is present, but it cannot work before we learn. So different reasons for which cystic fibrosis can occur. Because the faulty CFTR genes are recessive, someone with one faulty allele and one normal allele is able to make enough of the CFTR protein to remain healthy. Okay, what does this mean? Let me explain you. Suppose this blue one is the correct one and this red one is the faulty recessive one. When cell division, transcription occurs, the bonds are broken. Okay, let's see, it should be black. When transcription occurs, the bonds are broken, and thus this part unwinded, and this correct part also have unwinded. So what happens? The genes, the proteins produced from the correct one it will work, but that produced by the recessive one will not work. So still there will be enough CFTR proteins on the cells produced by this blue one, the correct gene, to help the normal functioning of the cell. But if both of them are recessive, then there will be no CFTR proteins to do that. So yeah, let's move on. The person is a symptom-free carrier of the disease. Each time two heterozygous have child, there is 1 in 4 chance that the child will have the disease. We know how this happens. Say this is heterozygous, means like both a dominant allele and a recessive allele is present. Yeah. So when they will form, like they will have a child, then the probability is this blue with this red one, why green? This is not actually in this chapter, but I'm still showing you if you have forgot. And then again the first, okay, let me write numbers so that I can explain you. 1, 2, 3, 4. So the first probability will be like 1 and 3. Now then again 1 and 4. Thus yeah, and then 2 and 3, and then 2 and 4. Thus, this is the probability of the genomes of their child. So the first child will have, the first child, by first child I mean this child will have 50% correct gene, correct protein, 50% wrong protein. The second child will have all of the right protein. The third child will have all of the wrong proteins. Thus, there won't be any transporter proteins of the chlorodynes to help the does the process of removing, helping to remove the mucus. And this child, again like the first child, will have 50% right protein, 50% wrong protein. Thus, they will be symptomless carriers. Okay. Because it is caused by a single gene, cystic fibrosis could be a good candidate for gene therapy because it is easier. Only one single gene is the problem. Okay, or like in single gene, 3 nucleotides. We can just insert the correct gene into a cell extracted from the alveoli and insert that into the person, and for some time the correct protein will be there, and thus chlorides can be like moved out of the cell, and thus mucus can be ejected. But it won't happen forever because the cells will die, and we have already learned that usually the cells cannot pass on that correct gene which we have genetically engineered to the daughter cells. Okay. If the normal dominant allele could be inserted into cells in the lungs, the correct CFTR should be made. In theory, there is no reason why this should not happen. In practice, there have been major problems in getting the allele into the cells. In the UK, trials began in 1993. The normal allele was inserted into liposomes, okay, which is then sprayed as an aerosol into the noses of the 9 volunteers. This succeeded in introducing the alleles into a few cells lining the nose, but the effect only lasted for a week because these cells have a very short lifespan. I've explained a problem that may happen because of the short lifespan. Researchers in the USA tried a different vector in a trial involving several people with cystic fibrosis. They introduced the normal allele into the normally harmless virus and then used these to carry the alleles into the passage of the gas exchange system. The allele did indeed enter some cells, but some of the volunteers experienced unpleasant side effects as a result of infection by the virus. The virus had caused infection. It did good, but then it did bad. It also caused infection, which was not our intended effect. Okay. As a result, the trials were stopped. To be used as a treatment, the allele really needs to get into many cells throughout the respiratory system, including the ones that divide to form the new surface cells. This has so far not been achieved because in therapy it is not treated. It is not permanently it is not a permanent solution. Thus, it is not a treatment. It's just a therapy. For permanent solutions to occur, like the correct gene has to be inserted into the cell, into its nucleus's genome, so that it can pass on to the daughter cells or even to the cells that produces in stem cells, or like the cells from which the alveolar cells are formed, so that all the next cells that will be produced after genetic engineering will carry the correct gene. More recently, a different approach has been taken. In some people with cystic fibrosis, the mutation in the gene has simply replaced one base with another. This has created a stop codon in the middle of the gene. The gene is transcribed in the normal way, but translation of ribosome stops when the stop codon is reached. This means that only a short length of the CFTR protein is made. A drug called PTC124 has been found to stop translation, to just keep going across this stop codon, so the entire protein is made, although with a wrong or missing amino acid in the middle of it. Even though there is a missing or wrong amino acid, at least if we produce the whole protein, then we could insert that one amino acid, 3 nucleotides, through viruses or retroviruses we have explained earlier. But if only 50% or 40% of the gene is produced, then it will be hard to to insert all of the genes and into the right manner. Okay. So, where was I? Clinical trials have shown hopeful signs that this may allow enough CFTR to be made to significantly relieve the symptoms of some people with cystic fibrosis. Sometimes, they may not have the 3 nucleotides absent, but the stop codon do not produce the protein. Thus, if we use that medication, then they will, since the translation will occur, but transcription will occur, and all of the protein will be produced, they may have enough proteins to survive. But it is much easier to do this than classic gene therapy because it only involves the patient taking a pill each day, because gene therapy is quite tough, and since if they do not have the 3 nucleotides nucleotides absent, if we only use the pill and the stop codon won't have its effect, then okay, normal gene is produced and we are taking pill each day and we can keep producing the protein. That's a good thing. Occasionally, DNA has been inserted directly into tissues without the use of any vector. The so-called naked DNA has been used in trials of gene therapy for skin, muscular, and heart disorders. The advantage of using this method is that it removes the problem associated with vectors, because we have seen that vectors, if there are vectors, then it quite produces few problems. Okay, somatic cell and germ cell gene therapy. Basically, germ cell is like the gametes. Gametes are, male gametes are the reproductive cells. They are haploid. They have half the chromosomes of a normal body cell, and male gametes are sperm, while the female gametes are the egg. And gametes are not like germ cells are not like only the gametes, but also like sexually reproduction cells. Basically gametes. Okay, and somatic cells are the body cells. So gene therapy involves introducing a correct allele into a person's cells as a treatment for genetic disease. So for all attempts to do this, humans have been replacing the alleles in body cells, otherwise known as somatic cells. However, another possibility would be to insert the allele into germ cells, that is, cells that are involved in sexual reproduction, such as gametes or an early embryo. For example, in theory, a woman with cystic fibrosis could opt to try to conceive a baby using in vitro fertilization, because she is the carrier of the diseased allele that causes the disease. Her baby there is a probability that the baby child may inherit that gene, and we have seen that the percentage. Okay, so what happens if it tries to in vitro fertilization? Let's see, eggs or oocytes can be harvested from her in the normal way. Then the correct allele for the CFTR gene could be injected into an egg, and this egg fertilized by sperm to produce a zygote. Thus the zygote will have correct gene, not the incorrect gene. At present, this is illegal in humans. However, it has been successfully done in other animals, although it looks as though there is a high chance of offspring produced this way having other unpredicted diseases. The problem that many people see with germ cell therapy is that all the cells of the child are produced from this genetically engineered zygote, and therefore will all carry the gene that has been inserted. When the child grows up and produces egg or sperm, these gametes will also contain the allele, and therefore it will pass on to their children. We said that the allele is in the germ line, being passed on from generation to generations, because of course, since the germ cells have the alleles, correct alleles, they can be produced over and over. They're in all of the cells, even the child's gametes or germ cells. So the germ line of the generation has been changed, because when that child will also conceive a baby, his or her child will also contain that genetically that gene for the correct CFTR. Yeah, just that.